Welcome students, and we are just going to have a continuation of another one short class, but it is very important for you to have this also one topic which I have to discuss finally for the QC SH, that is for example quality control of herbal drugs students who have taken as an elective subject in 8th semester. And this we have discussed previously also. We had a discussion before I hope in your previous maybe last first session itself we had markers, analytical markers and active markers, like that we have just had a discussion. But this is somewhat a little some examples and details I have added about the role of chemical and biological markers in standardization of herbal products. Let me make this one little more clear to show you. And see here, it is about the role of chemical and biological markers in standardization of herbal products, and that's what we are going to discuss. It's not a big one, but it is very important actually. It is needed for you to give the examples, and with more you can just refer and you can read, and it will be interesting for you in the future. It has some new importance in about the markers, especially the role of chemical and biological markers in standardization of herbal products is our topic today. And type of marker compounds: one is chemical markers, two is biological markers, and the third one is molecular or DNA markers. So the chemical markers, number one, the chemical markers are an unique component that contributes to the therapeutic effect of herbal medicine. It contributes the therapeutic effect of the herbal medicine. Understand? And the study of chemical markers is applicable to main research area, including the authentication or authenticating the particular plant or a genuine species, whether it is a genuine species which was selected for your research, and the search of new resources or substitutes or raw materials, optimization of the extraction and the purification method, structural elucidation, how important for the chemistry, structural elucidation and purity determination. So these all gives important for the chemical markers. And what are the type of chemical markers? If you see, an active principle, and I say it is a chemical marker, active principle, and it is active principle, or you can say active principle chemical marker. Well defined chemicals with known clinical activity. Well defined chemicals with known clinical activity. For example, methanolides, which is present in Withinia somniferum, it has an antifungal activity and it is having more psoriasis. We are always depending on Withinia somniferum. So it is a well-known chemical, and a clinical activity is also known. It is used for antifungal and it is used for psoriasis. So you can say active markers, again active principle, active markers, which is active marker, chemical marker. These both are slightly interlinked. So you should have a small difference is there. That is, I am telling that it is a well-known clinical activity is known, like methanolides is told as an example. Here they are, that is active markers, which we say the second one, they are also constitute a group of constituents which is known pharmacological activity that contributes to efficacy. But the line which underline you should remember, that is may or may not be proven clinical efficacy. May or may not proven clinical efficacy. There we told with known clinical activity. That is the only difference. Example, you can say allicin is present in Allium sativum. We know that Allium sativum is a very common, which is the we are using for the food and everything, and it has more medicinal activity. But in that you can see allicin is present, and it may or may not be proven clinical efficacy, but it is more useful. That is called an active marker. Okay, so active principle and active markers. Active principle, it's also a chemical marker. Active marker is also a chemical second one. So they both are interchangeable. You should write an example like this. An analytical marker, we know that the chemical markers, they are constitutes a group of constituents that serve solely for analytical purpose, and they have no clinical or pharmacological activities. They don't have any clinical or pharmacological activities. They help in identification of raw material, and raw and extracts are used to achieve standardization. Example, alkyl amides. Alkyl amides, you see, alkyl amides is a common general name, alkyl amides. We cannot say alkyl amides. There are many different types of alkyl amides, which we cannot say specifically the pharmacological action or therapeutic activity, we cannot say. So, alkyl amides found in the roots of Echinia purpurea, Echinia angustifolia. But you can see totally it is absent in Echinia pallida. So this is three of them are very good, beautiful flower plants. So this they have identified to just an example they have given. So this is an example which two of them they have, one of them is not, one of the species it doesn't have. They all are having different, same type of flower, color and flowers, which is very difficult to identify. Only thing is by means of chemical markers or analytical markers, it is possible for to differentiate the species. Understand? That is an example. So this is just I want to show the picture of the there is Echinia purpurea. See the color. It is on Astraceae family, and beautiful color. It is also present in it's usually commonly available in the United States, and we have in the garden. And you see this is the Echinia angustifolia. The same flower, just as flower color is slightly different, but even it can be purple also slightly. And you can see it is also a cone shaped flower, or you can say black salmon Echiniaceae, and it is a North American plant species of sunflower family, and it is widespread across some of the great plains of central Canada and central United States also even, and additional population in nearby region. So this is one, and the third one which you saw, it's not containing, especially the alkyl amide is not present. Other two they have the alkyl amide. That is what analytical markers we are telling the example. Can you whether in the picture you can write the example. That is why I want to say about to the students to show some examples. If you write, it will be easy it will be interesting for the evaluator, and they will give full mark. If you write botanical source and family, because when you are writing about pharmacognosy, it's necessary for you to write about botanical name and botanical source and family, and even the constituents with chemical structures is very, very important to know about. Because you are a chemistry background pharmacy students, they will expect you to write about structures and the botanical names. And Echinia pallidae is an Astraceae family. It is also, you can see the color of the plant, and just a change in the corolla of the flower is having a difference. So the roots of this plant, they have identified the presence of alkyl amide. Understand? That is what they can do as an analytical marker. So these three markers you can just differentiate. And fourth one is negative marker. It is also a chemical marker, differentiate negative marker. Negative marker demonstrate the allergic or toxic properties. Example, ginkgolic acidin or ginkgo showed signs of apoptosis as well as necrosis. So this is an example. That is ginkgolic acidin is an example from ginkgo plant or ginkgo plant, which we are isolated and we are doing this marker. It is also a chemical marker, but it is a negative marker, demonstrate allergy and toxic property. Toxic property, it is opposite, which is a toxic properties can be identified. That is why it is called negative markers. Okay. And application of chemical markers to identify the adulterant and to identify the components by means of presence, and we have given the example also. Differentiation of the herbal medicines with multiple sources, and also succeed determination of best harvesting time. And you can see I have just mentioned that is C there, and D it should be confirmation of collection of site, and E is assessment of processing method. Understand? This is what, and you can see there is a issue in this. Instead of making it, I will make it clear. Here it is the C, and this is, we can make it as a a D, understand, and why you have to make it, and this is as E, and this one should be F. Okay, and it's possible. It's just I want to show why, because the student should know that it's an easy thing which we can do in presentation, because you have to present your project work and everything. So you should be take it very coolly when you are present. If there is any mistakes in your slides, you should be able to explain it. Understand? So identification of adulterants, differentiation of herbal medicine of multiple sources, and determination of best harvesting time, and even confirmation of collecting site, even it can be determined by means of using chemical markers, and assessment of processing methods, and identification of quantitative determination of proprietary products. And if you see, to evaluate the stability of proprietary products, quality over time, and determination of recommended shelf life, storage, hold on, all these things can be studied by means of these chemical markers. And diagnosis of herbal intoxication, in which toxic components may be used as chemical markers. In screening methods, rapid diagnosis of acute hidden aconite poisoning in urine analysis. You see, example we are given, diagnosis of herbal intoxication is in which toxic components may be used to chemical markers. So example, you can say rapid diagnosis of acute hidden aconite poisoning. In a urine sample we can take, and by means of HPLC mass spectroscopy, we have discussed about these two instruments linked with, for example, high performance liquid chromatography connected with the mass spectroscopy. We can easily identify which compound is present in that. It's possible for you to separate and to identify with using a chemical marker. Detection of leading components of new drug delivery. So you can say gambogic acid. You have to give the example. Gambogic acid is a potential lead compound for a new anti-cancer drug. So it's just an example which you can give for a new drug delivery system can also be applied for by means of using chemical markers. And now we are coming to the biochemical markers. What do you mean by biochemical markers? Biochemical markers are either proteins or isozymes. Isozymes are known as allozymes or enzymes that are functionally similar but differ from each other in the location. So structure, net charge, electrophoretic, mobility, type of activators and inhibitors, and heat stability. I am repeating for you. It's an enzyme, allozymes, and what is the main location? You can say differ in each other by means of location. Structure, I did not bold it actually. Location alone I have bolded it. You can see it should be bolded actually. You see, structure, net charge, because it's a proteinous nature. It has nutrients and charge, electrophoric mobility towards the charge, and type of activator and inhibitors and heat stability. This you have to write about the biochemical markers. The isozyme pattern has been used in many population genetic study, including measure of outcrossing rates and subpopulation structure and population divergence identification. You see, we were seeing about we need the DNA structures and DNA fingerprinting, all these things. You can identify certain the chromatograms and the DNA fingerprinting will give you the specificity, and it can easily identify which one belongs to which group. And we know that. What do you mean by DNA testing and all? So just you should know that how it is, what do you mean by biochemical markers and how this genetic studies can be carried out. These are These are particularly valuable to distinguish closely related species. Very closely related species can be identified by means of this type of biochemical markers, and therefore useful to study diversity and identification of the the crops. You have to write this very clearly. Useful to study diversity and identification of crops. Example, you see the example which I have given, the isozymes pattern of glucose 6 phosphate. Glucose 6 phosphatide, you can say, glucose 6 phosphatide hydrogenase is the enzyme, has been used for the identification of Eclipta prostrate false daisy, that the flower is false daisy flower. False daisy is the name of this plant. And the peroxidase isozyme pattern is used for distinguishing the three types of Gastrodia elata variants. Just you can write the name of the plant, because I am not going for the details about that much details. Just I am giving, I am having the tendency to even to read. For me also, I want to know about that. But our syllabus does not say that is you have to go for the details about the genetic activities whichever happen there. Just you can write the examples. Understand? Can you follow how they are using for the false daisy and how they are using for the Gastrodia, elata, and the variants. The three variants have been used by the peroxide isozymes. And molecular and genetic markers are DNA markers. The third one, molecular or genetic markers are DNA markers. It's a specific, just coming next specificity in the DNA. So DNA markers are most widely type of markers, mainly due to their abundance. DNA markers are unlimited in number and are not affected by environmental factors, age, and the physiological condition of the plant. So there is a big advantage. It doesn't affect the plant age or any environmental factors. So that's why DNA markers are having more useful. The DNA can be extracted from fresh or dry. Fresh also you can use, or a dry plant also can be, tissues can be used for isolating the DNA. Hence the physical form of the sample for assessment does not have any restriction in detection, because fresh or dried can also be used for your sample to take a DNA. Understand? So that's why the molecular or genetic markers, or it is called as DNA markers. So you can see the application of DNA markers. Number one, in herbal drug technology, in our subject, DNA based molecular markers has proved their utility in the field like taxonomy, physiology, and embryology and genetics. You can write very clearly what is its application. It is very much applicable for taxonomy and physiology and embryology and genetics. And genetic variation, genotype, random amplified polymorphic DNA based molecular marker have been found to be useful, that is RAPD, have been found to be useful in differentiating different assessing of, example we have given, Andrographis paniculata, Astracita indica, and Allium coenoprasum. Yes, silent, you can say coenoprasum. Collected from different geographical location. They have done RAPD is very much useful, random amplified polymorphic DNA. So you can write an example, genetic variation of genotype can be identified by means of RAPD with an example of Andrographis paniculata, Astracea indica, and Allium coenoprasum. Understand? Coenoprasum can be given. So in interspecies variation, next one, interspecies variation has been identified by means of using restriction fragment length polymorphism, restricted fragment length polymorphism, RFLP. Please, you have to write this. It's very important. And what is the example which can be used for we can use, differentiate the genera. Glycerista glabra, Curcumbellanga or curcumin, and Echinaceae can also be gone for this interspecies. variation can be identified. Understand? And authentication of medicinal plants, rare, the expensive medicinal plants, especially expensive medicinal plants, usually they are adulterated. They are adding substituted they used to do for a marketing purpose, for a business purpose. So that can be identified. Morphological similarity, and they will be morphological similar, and they will see looking very similar. They used to add. So easily available and less expensive species. So they will add less expensive species in the high expensive species which they want to sold for a. So in that case, sequence characteristic, that means which you have seen amplified region, that means sequence characterized amplified region type, or polymerized chain reaction, or random amplified polymorphic DNA, or restricted fragment length polymerism, are used for differentiation of the plant and to detect substitution or detection by other close related species. So these all abbreviations is needed for the student should know, because you know PCR test, now you are doing for very PCR test, antigen test, like that you are very common in talking about the corona test you might have done. So polymer change reactions can also be used for differentiating plant species in different, like by species can be identified. Sequence characterizing amplified region, polymer change reaction PCR can be used, and random amplified polymorphic DNA, and restriction fragment length polymorphism can also be used for differentiating the plant and to detect substituent, adulteration and other closely related species. Just I have bolded it, and it is very authentication of a medicinal plant is very, very important. Example, Sweetia cerata. Sweetia cerata is frequently adulterated with the substituent of Andrographis paniculata, can be identified by means of any type of this DNA marker can be used to identify. Medical plant breeding also they are using by means of DNA markers. So how the molecular markers are has been as a tool to verify sexual and apomictic offspring of intraspecific means the same species across in Hypericum perfratum, Hypericum perfratum. It can be either St. John's wort, is the plant's name St. John's wort. We have seen this plant actually, yellow color flower, beautiful flower. It has a lot of medicinal uses as well as known as diuretic and anthelmintic drug it is. It can be food and nutraceuticals also. The molecular markers, DNA markers, capsicum, maize and soybean have been successfully you can say, successively you can say, characterized by means of non-GMO products. What do you mean by non-GMO? Because non-genetically modified organism, that is GMO means GMO here, non-GMO means it is not genetically modified organism. GMO means genetically modified organism, the novel drug related to laboratory using genetic modified engineering technique. Just I added for the definition, understand? And as a new pharmacognostical tool, DNA markers have shown utility in quality control of commercially important botanical like ginseng, Andrographis paniculata and Echinacea. Although DNA analysis is currently considered to be innovative technology, it has been certain limitation due to which its use has been limited in academia. So another important issue in DNA fingerprinting will remain the same irrespective to the plant part used, while the phytochemical content will vary with physiology, plant parts and used and environment. Okay, so that's very, very important topic which we have discussed. Kindly please go through it, and it's a short topic, and the markers which already you have seen, and this markers with example. Kindly I request the students to go through it. Thank you very much for being with me.